Hello out there, Virtual Evolution. My name is Alex Skills and I'm a postdoc in the Landscape Ecology group at WSL and ETH Zurich. Today I'm going to be talking about the role of temperature and life history dependent speciation rates in shaping biodiversity. I'm going to start off today by telling you all something that we know, and that's that environmental energy, whether measured as solar radiation, temperature or productivity, is a really good predictor of biodiversity, and particularly at broad spatial scales. Something we don't know, however, is why, but there are many hypotheses. So today I'm going to be talking about one of the major hypotheses linking energy and biodiversity. This is the energy and evolutionary rates hypothesis, or sometimes known as the evolutionary speed hypothesis. The hypothesis suggests that organisms in high energy environments have faster mutation rates, either due to more UV damage or oxidative damage from faster metabolic rates. These organisms also have faster life histories, so that's a suite of traits that includes small body sizes, fast growth rates, and shorter generation times. This is suggested to increase rates of molecular evolution and the rate of selection and adaptations in populations that live in warm places, which will increase the formation of reproductive isolation and therefore rate of speciation. This hypothesis has been tested typically by finding correlations that support links in this pathway. For example, a classic macroevolutionary approach to this would be to look for a link between substitution rates and diversification rates. One problem we face with these kinds of analyses is that as we've come to learn a lot about the complexity of these different mechanisms and how they interact, we know that it's not always possible to draw inferences about processes by looking at patterns. One way to bridge this gap is using process-based models. So these are models that explicitly incorporate specific eco-evolutionary mechanisms. And there have been lots of recent advances on this front in the development of spatial simulation models, which allow us to incorporate complex spatially dynamic processes more directly into macroevolutionary models. The aims of this study are threefold. First, to use process-based spatial simulation models to understand how biodiversity patterns emerge over time under the energy and evolutionary rates hypothesis. Then we want to see if any biodiversity patterns are diagnostic of this hypothesis, to see if we can make predictions in real data. To do this, we use a spatially explicit simulation engine, Genesis. I'll go into the model in a little bit of detail here, but I recommend anyone interested in the general framework to go and see Oscar Hagen's talk here at Virtual Evolution, or to check out the preprint on bioRxiv. Genesis is a grid cell-based model that follows the diversification dynamics of populations across a dynamic landscape through time. The model is modular and can be modified by the user to specify different processes directly in order to model and compare alternative variations of different evolutionary models. This allows us to incorporate real paleoenvironmental reconstructions into the model to assess how diversification dynamics have played out in conjunction with environmental and plate tectonic changes. In this study, we used temperature and aridity reconstructions over the past 65 million years of the Cenozoic period as inputs. In interaction with this dynamic landscape, we model four main processes, which interplay at discrete time steps and lead to emergent patterns such as species richness, which you can see here in the animation on the right. So the model includes dispersal. So this is the kernel of movement that populations in each grid cell can take at each time step, and it facilitates movement across the landscape. We also model trait evolution, where species have an environmental niche trait, as well as a body size trait, which is taken as a proxy for life history. And these traits evolve under Brownian motion. We also model ecological interactions, where the presence, absence, and abundance of populations in grid cells is based on how well adapted populations are to the environment, as well as the presence of other species. Finally, we also model speciation and population divergence. And I'm going to go into this in a little bit more detail now, as it's key to the energy and evolutionary rates hypothesis. In Genesis, speciation is explicitly allopatric, and populations of a species, here I and K, diverge genetically from one another when they become separate at time alpha. Divergence then occurs at rate lambda, and speciation occurs once divergence has crossed a threshold S. At this point, populations are considered sufficiently divergent to be considered new species, and they evolve independently. And in this study, we implement four different submodels in which the rate in which populations diverge, so lambda, varies. So under M1, lambda is a function of temperature. Under M2, lambda is a function of body size. Under M3, lambda is a function of both temperature and body size. We compare these three models to a null model, M0, in which lambda is independent of both temperature and body size and is drawn from a uniform distribution. So to reiterate our aims in the context of this model, what we really want to know is what biodiversity patterns emerge under these four different submodels. Are there any biodiversity patterns that might be diagnostic of these different models? And then we want to see which models might be best supported by real data. The Genesis model gives us heaps of information as output, which is directly comparable to commonplace biodiversity data. So this is species distributions, their phylogenetic relationships, and traits. From the spatial, phylogenetic, and functional information, we extracted 50 different summary statistics. And some corresponding examples include things like the correlation coefficient between temperature and species richness, phylogenetic tree imbalance, and skewness of the body size frequency distribution. From these summary statistics, we used a classification machine learning approach to see if the different submodels of population divergence led to distinctly different distributions of summary statistics. So we used a k-fold cross-validation approach to train different classification models, including random forest and discriminant analysis. Here we basically train a model on a subset of the data and try and predict the correct submodel on a withheld subset. We can then estimate the reclassification accuracy under these different methods. So then to perform model selection, basically what we do is we get empirical data in the same format as our simulations, and we estimate the same 50 summary statistics. We then take the best performing model and try and estimate the submodel of population divergence from those summary statistics. So now jumping straight into the results, our first aim was to see if biodiversity patterns differ between these submodels of population divergence. The reclassification accuracy of the machine learning methods are a direct measure of this. So highly different patterns would lead to a really high reclassification accuracy. We found using a range of these machine learning methods, we were able to reclassify the correct model in between 70 and 75% of simulations. This is considered a substantial model accuracy, suggesting that biodiversity patterns do differ between models of population divergence. This brings us to the next aim, which is to see which patterns are diagnostic and informative of these different submodels and these different processes. So to investigate this, we can look directly at the contribution of different summary statistics to the ability for the model to discriminate between submodels using variable importance factors. Some summary statistics have very high values for this. For example, a correlation between species body sizes and diversification rates estimated using TIP metrics is very good at separating models with body size dependency of population divergence, which makes a lot of sense. Other summary statistics, on the other hand, are not informative at all and do not help us discriminate between submodels. For example, a correlation between latitude and species richness, known as the latitudinal diversity gradient, emerges under all four models and seems to be independent of the model of population divergence. So this suggests that broad-scale spatial diversity patterns are not very informative of underlying processes. Our final aim was to see which model was best supported by empirical data. So to do this, we estimated the 50 summary statistics that match our simulated data. We did this in 46 different clades of vertebrates at the level of order and infraorder of squamate reptiles. So what we found was really strong support for M1, which is a temperature-dependent model of population divergence. In most clades, we found this, and in a further 9 clades, we found support for M3, which is temperature and body size dependent rates. So that means in total, 87% of clades of 40 different clades had greater support for temperature dependency in population divergence rather than temperature independent models. So what does it all mean? Well, the take-home messages are that small changes to the model of population divergence lead to measurable differences in biodiversity patterns. Using multiple biodiversity metrics simultaneously helps us fit complex models to data, and that temperature-dependent rates of population divergence are best supported by real data. So thanks everyone for your virtual attention. I'd just like to thank Wilhelmina Bach, Oskar Hagen, Loic Pelissier and Walter Jetz for co-authors on this study, as well as Benjamin Fluke for technical support, and both the Moritz Lab and Macroevolution and Macroecology group at the Australian National University for feedback. Cheers.